Okay, hi everyone, welcome. Today I want to talk about inflammation and therapeutics. So there's this central idea in infectious disease that we've discussed, that there is a massive immune component in a range of different infectious diseases. So the way in which many causative agents of disease actually initiate a disease state is via immune activation. So the immune system activates in response to the pathogen, and this immune system activation causes host damage. It's really a central concept stretching throughout infectious disease, and it's one of the reasons why a range of different therapeutics targeting infectious diseases will actually directly inhibit the immune response. Specifically, we tend to think about inflammation as being really important in this process. So inflammation is again a consequence of the immune response. So when the immune system detects microorganisms, it initiates inflammation, and some of the characteristics of inflammation, like edema, so swelling, can be causative of specific pathologies. So what we want to do now is understand exactly what this immune response is, and you'll delve into this in a lot of detail in clinical immunology module that you'll do. So that's again got this big immunological focus, but to really understand infectious disease and specifically some of the therapeutic aspects, we also need to understand exactly what inflammation is, because there's this idea that immunology and in the study of infectious disease, so portions of microbiology, are really just two sides of the same coin, because when an infection is occurring, it's occurring in the context of the immune system. So to fully understand infectious disease, we need to understand the immune response, and specifically the part of the immune response that is most interesting and most relevant to the ability of the immune system to contribute to pathology is inflammation, because as we'll see, and as we've actually explored in the module, and I'm sure you've explored in your undergraduate degrees, there are many pathologies associated with infectious agents that are involving inflammation. So what we want to do initially is make sure we understand the immune response that's occurring here. So obviously immunology is a massive field and we don't have the scope to cover it in any kind of comprehensive way now. But what we can do is look at the specific pathway that a lot of different therapeutics target. And again, we're focusing specifically on inflammation because it is very important in the context of the immune system's ability to contribute to infectious disease. So we're going to look at specifically inflammation and the innate immune pathway activation that leads to inflammation. And just a little bit of a reminder in terms of immunity, we're talking about this massive complex immune system that the host has, and we can generally split portions of this complex immune system into two camps. We can say there is the adaptive immune response, the adaptive portion of the immune system, and the innate portion of the immune system. Now there's obviously massive overlap here, but it makes sense to split the immune system into these two arms just for our understanding, and because it does work in practice if we also appreciate there is this huge overlap. I'm sure we know what the adaptive immune system is. What we're talking about here is the innate immune system. So the innate immune system is essentially the portion of the immune system that is non-specific. So it launches a non-specific antimicrobial response as a response to the presence of a range of microorganism features. So it's not launched to target a specific microorganism. It can be launched in response to a specific microorganism, but the response is a general response and it's initiated by general features on the microorganism. So we're not really thinking about antibodies here. We're thinking about receptors that can detect microbial features, and this broad, wide-ranging immune response is initiated as a consequence. And this is one of the reasons why this process, which we will define the end point as being inflammation, is so problematic in many pathologies, because it is this broad sweeping response. It's not targeted against a particular microorganism. It's just a change in the physiological state of the host, and this change can again be causative of pathology. And again, we have that information reiterated here. So when we're talking about innate immunity, we're talking about this widespread, non-specific response that is designed to clear any microorganisms that are present within the host. And the nature of this response, being widespread, being broad, is problematic, because again, it can contribute to some pathologies. Okay, so as part of this immune response, we need to understand the induction. So we've said this innate arm of the immune system is induced in response to microorganisms. So there must be some recognition of microorganisms occurring. That's how the induction is triggered. And this occurs when components of the innate immune system recognize specific features present on microorganisms. Now, these features are called pathogen-associated molecular patterns, and generally speaking, these features are present on a wide range of microorganisms. So this isn't a specific recognition of a particular type of microorganism, certainly not a particular species of microorganism. It's a recognition of common features that are found on a range of different microorganisms, and the induction of a broad immune response as a consequence of this. So we can hopefully see already how it's not a specific response, and we can see the characteristics of the response reflecting this, because it's not specific recognition. Again, the features that are recognized are pathogen-associated molecular patterns. That's how we refer to them, and we'll look at some examples of these as we work our way through the lecture. Now, we need to think about what a PAMP actually is in terms of what its characteristics are, and its characteristics again reflect the nature of innate immunity as a broad response. So this arm of the immune system has evolved to recognize PAMPs that fit some specific criteria. And the reason for this is, well, it's manifold, but one of the reasons for this is because it needs to be activated. So this innate immune response needs to be activated by as many different microorganisms as possible. It needs to be as broad a response as possible in terms of induction, because this is the nature of the response. It should be induced by any microorganisms that are present, and that's what we see. So the way the system has evolved is it recognizes features on microorganisms that are present on a large number of microorganisms typically, because again, this innate immune response needs to recognize as many microorganisms as possible. And one way it can do that is it has evolved to recognize features that are present on many different microorganisms. So recognize common features on different microorganisms. PAMPs are also easily distinguishable from host, and what this means is the features that the innate immune system have evolved to recognize don't look like host cell features. And this is, fairly obvious why this is the case. It's because if the innate immune system is capable of recognizing microbial features that appear similar to host cell features, then this immune system arm could potentially be recognizing actual host cell features, and therefore inducing an immune response in response to the host's own features, which is obviously a massive problem, because that's what autoimmunity is, and that can be causative of really serious disease in and of itself. The other criteria that these PAMPs fit is that they have to be essential for pathogen survival. So pathogens mutate rapidly, and the rate is different dependent upon the specific group of microorganism we're looking at, but compared to host cells, the rate is always very rapid, and there is therefore the likelihood that microorganisms due to mutative rates and also reproductive rates, are able to evolve to lose or significantly change the feature that the immune system is recognizing. So if the innate immune system has evolved to recognize a microbial feature that is different from the host cell features, so it meets one of the criteria, and it's present on a wide range of microorganisms, so it meets the other criteria, the microorganism could potentially quickly evolve to lose that feature or to change that feature so significantly that the innate immune system doesn't recognize it anymore. Now, as a kind of solution to this, the innate immune system has evolved to recognize PAMPs that are essential for pathogen survival, so PAMPs that the microorganism can't easily discard without a massive fitness cost or can't easily change for various reasons. So this is what the PAMPs are. They fit these criteria. They are microbial features that the innate immune system has evolved to recognize. And these PAMPs induce this innate immune response, which is the first stage of inflammation, which again is very often playing a role in pathology. So if we look at some specific examples of PAMPs, hopefully we'll see how these meet the criteria, these features. So foreign nucleic acids, this is a massive one for viral infections, so viruses. So many viruses will have genome compositions that aren't normal nucleic acid formats that are seen in a healthy cell. In addition to this, many will produce products of genome replication or genome expression, so genomic products that are different to what would normally be seen in a host cell, and these can be really potent inducers of the immune response, because again, these are specific nucleic acid types that are not normally seen in a host cell, so they are strong indicators of an immune response. They're easily distinguishable from host because again, they're types of nucleic acid combination that aren't seen in a healthy host. They're essential for pathogen survival because they're part of the microorganism's genome or the replicative or expressive process of the genome, and they're present on a wide range of pathogens. So all the different types of nucleic acid combinations that are recognized by the innate immune system are present in a range of different viruses. So if we think about the specific nucleic acid combinations that we see, double-stranded RNA is a huge one. So some viruses have double-stranded RNA genomes, or they have a stage in the reproduction of their genome or expression of their genome that involves double-stranded RNA. This double-stranded RNA can induce a really potent immune response because there should never be double-stranded RNA within the host cell. It's not normal, so it meets the criteria to be classified as a PAMP, and the immune system has evolved to recognize it as such. DNA RNA complex, so in a normal healthy cell it would never be seen that DNA is bound to RNA. However, in the replication of some viruses, retroviruses for example, so reverse transcription, one product or intermediate rather we should say, because it's made during the process, it's not the end product of reverse transcription, but that is RNA bound to DNA. So the immune system has evolved to recognize RNA-DNA duplexes because again, it meets the criteria outlined on the right. The next is cytoplasmic DNA. So in a normal healthy cell, there shouldn't be DNA in the cytoplasm. DNA should be in the nucleus. If DNA is detected in the cytoplasm, this indicates maybe best case scenario, it's leaked out of the mitochondria, so the cell is significantly damaged. However, what it is seen to be interpreted as by the immune system is an indicator of microbial presence, because some viruses will replicate in the cytoplasm. So if there's DNA in the cytoplasm, this is an indicator of viral presence. So there are various bacterial features as well. So LPS, lipopolysaccharide, and flagellin, these are found on bacteria and they're never found in a healthy cell. They're actually found in a wide range of bacteria. So in LPS, as I'm sure we know, we've studied undergraduate microbiology modules at some point. LPS found on a massive number of bacteria, and flagellin found in bacteria that have flagella. So these meet the criteria on the right. Structural proteins found on viruses, fungal cell wall carbohydrates, and GPI anchors on protozoa as well. So these all meet the criteria on the right. Distinguishable from host, essential for pathogen survival, and present on a wide range of pathogens. So what occurs in the innate immune system is pattern recognition receptors recognize these PAMPs. So pattern recognition receptors are the receptor component. So these are the component of the innate immune system that actually recognize the PAMPs. Now you'll sometimes see in the literature the term TLR being used. TLR means toll-like receptor, and toll-like receptors are a significant group of pattern recognition receptors, so a group of PRRs. TLRs and PRRs are not synonymous terms, but sometimes it almost seems like they're used that way because a large proportion of PRRs are TLRs, but there are other PRRs as well. There are cytoplasmic PRRs that recognize, again, that cytoplasmic DNA. TLRs tend to sit in the membranes. Okay. We have 9 different TLRs, and these recognize different PAMPs, some of which we've just looked at. So on the right we have an example of activation where TLR is being bound to poly-IC, which is poly-IC is a mimic of a nucleic acid combination that triggers innate immune activation, so something that we just looked at. When the TLR is activated, there's downstream activation of transcription factors like IRF3 or NF-kappa B, and these transcription factors then activate a range of different signaling molecules and proteins to be expressed. So there are these changes in host cell expression, and these changes are a downstream consequence of the activation of pattern recognition receptors. And it's important to note there's a significant amount of convergence. So regardless of which pattern recognition receptors are activated, there's massive overlap in the transcription factors that are activated. So there's overlap in the process overall, and the endpoint is often very similar. So these transcription factors include IRF3, interferon regulatory factor 3 and 7, and also NF-kappa B. And again, I'm sure we know what transcription factors are. They're activated, and once they're activated, they move into the nucleus where they induce the expression of specific genes. In this case, so in the innate immune system, this involves signaling molecules, which are interferons, cytokines, and chemokines. And once these are produced and released, they induce widespread changes throughout the immune system. Okay, so here's an overview of that process. So PRRs are present on cells, they detect, or in cells, they detect these PAMPs. Transcription factors are then activated as a consequence of PRR activation. Interferons and cytokines and chemokines are produced as a consequence of transcription factor activation. And then these signaling molecules induce widespread changes throughout the immune system, throughout the body, that are antimicrobial. So they are antimicrobial changes. So they are induced to fortify the body against infection, but they also have negative consequences potentially because these signaling molecules, interferons, cytokines, and chemokines, are inducing inflammation. That's how we define this antimicrobial state, and there are, again, pathological consequences of inflammation in many cases. Okay, so now what we want to do is think about how these processes can actually be manipulated therapeutically. So the first drug we're going to look at is baricitinib, and we're going to see how this drug essentially intervenes on the process we've just identified, and in doing so, hopefully reduces the pathology of a particular infectious disease. Okay, so it initially attained emergency use authorization for for the treatment of COVID-19. Again, we have the signaling pathways that we've discussed here. We have activation of PLRs, which induces activation of transcription factors, and these transcription factors activate the induction of various signaling molecules, so interferons, cytokines, chemokines, and these signaling molecules induce a protective response throughout the immune system, which includes inflammation. Okay, now what baricitinib essentially does is it counteracts the signaling of one really important signaling molecule that is induced as part of the pathway we've just looked at. So we mentioned interferons, chemokines, and cytokines. Interferons are massively important here. It's a reason why they're listed first. Interferons are really the primary cause of inducing inflammation. And if we look at COVID-19 and also other infectious diseases, we have often again see that inflammation is playing a central role. And again, this is what baricitinib is doing. It's inhibiting the action of interferon, because interferon is a signaling molecule, and when it's released, it will induce downstream signaling pathways of its own to actually enact the changes. And one of these pathways is the JAK-STAT pathway, which interferon again activates, and baricitinib counteracts this activity. And here's a separate study looking at the usefulness of baricitinib in combination with remdesivir. So remdesivir is a drug we looked at in a paper yesterday, and it's an antiviral. Baricitinib is a different type of drug, isn't it? It's targeting the immune system, and they're used in combination. And this is something we'll often see in papers discussing viral infections, that there are two facets to approaching treatment. One is to actually inhibit the virus itself, so antiviral therapies, and the other is to target the immune system, so again often to suppress the inflammatory response, which is happening here. Okay, so that's baricitinib. What we want to do now is look a little bit more at interferon. So interferon is quite complex when we're thinking about a therapeutic perspective from infectious diseases. So it can seem intuitive on the surface that because interferon induces an antimicrobial state, it could be used potentially therapeutically to treat viral infections. And obviously, as we know, as we've worked through this lecture and we've talked about on the module, a lot of the time the relationship between the immune system and the infectious disease is a bit more complex than that. And so you probably won't actually be surprised to hear that interferon isn't particularly useful as a treatment for some viral infectious diseases. It can be useful in some contexts, and you can look at the literature on that, but it's complex. It's a really complex story. If we think about HIV specifically, it was initially thought that interferon could be used to control HIV in individuals long term. And again, the idea was interferon will be inducing an immune activated state, so inducing a protective state that will reduce the replication of HIV and will therefore stop the spread of the virus through the body, and this will ultimately prevent progression to AIDS, which is the goal of HIV treatment when it's a suppressive treatment. The problem, however, is that as we've looked at, the innate immune response is complex, and the inflammation associated with the innate immune response can often be causative of pathology. The innate immune response is designed to be a rapid, almost immediate response to the presence of microorganisms. It's not designed, evolved to be a chronic state. And chronic immune activation is often a problem, and it's a problem associated with chronic infectious diseases because often the immune system is in a chronic state of inflammation. And this is what's seen when interferon was used in the context of HIV infection. It's seen it wasn't as effective as was hoped, and there's some specific concerns in the context of HIV infection. So firstly, as we've explored, we can consider that maybe it's not the best idea to be inducing a constant state of immune activation, particularly when we're talking about innate immunity, which has evolved as a short-term immediate response to the presence of microorganisms. But if we think about HIV specifically, we know the progression from HIV to AIDS occurs when there is a loss of immune cells, and this loss of immune cells occurs due to the virus infecting immune cells and causing their death. However, there's more to the story, because the activation of the immune system in the case of HIV actually causes a more rapid loss of immune cells. So what is essentially occurring when the immune system is activated in HIV is there's a really rapid turnover of CD4 plus T cells. So these CD4 plus T cells are being activated by the immune system, and as they're being activated from precursor cell types, HIV is able to infect these cells. So by constitutively activating the immune system, we can be inadvertently creating a large pool of cells for HIV to infect. And long term, the precursor cell types are not, and they are a finite supply. Okay, so they can't, so the immune system can't continually produce CD4 T cells at a rapid rate, particularly if there is a high turnover with the virus depleting these immune cells from the immune system. And also, if we consider these immune cells have a half-life, if you like, anyway, they don't last forever, they will die naturally after a specific period of time. So if there is this rapid rate of turnover combined with a really high immune activation, then very quickly this can significantly contribute to the loss of these immune cells long term, because the immune system just can't keep up, it can't replenish the CD4 T cells. And this is actually what we see when we try to understand HIV infection in detail. We see that yes, the virus does infect immune cells, and this does cause lysis of the cells, because again, HIV steals bits of the membrane when it buds off, and also because an infected cell is put under a huge amount of strain, because it is essentially being forced to produce viral proteins, and this means it is not capable of sustaining normal cell functions. So this all happens. However, immune activation is also really, really important and a significant contributing factor. So that's not to say, by the way, that interferon doesn't have a role in HIV treatment. It's just to say that the situation is a little bit more complex, or quite a bit more complex, than just utilizing interferon as a way to induce a protective immune response that therefore reduces the progression of disease. And here is a really strong and useful review that essentially discusses this idea of interferons and HIV infection, and discusses the complexity of the relationship between the two.